Hello everyone. In this video we're gonna talk about about glycolysis. So the video will be divided into two major portions. In the first stage we will discuss the overview of glycolysis just to get a general idea of it. And the second part of the video will focus on describing the stages of glycolysis and their significance. So what is glycolysis? I'm sure you would have heard this name quite a bit till now, and that's why you want to just know more about it. So, glycolysis is a energy yielding pathway which is virtually present in all cells, both prokaryotic and eukaryotic. And this pathway takes place in the cytoplasm of the cell. So basically, glycolysis is breakdown of glucose in by series of 10 steps pathway, which basically gives you ATP, which is the currency of energy in the cell. So glycolysis can be glycolysis can be uh, thought of as compromising two stages. So let's say glycolysis stage 1 and stage 2. The stage 1 of the glycolysis begins with breakage of glucose till the step when fructose 1,6 bisphosphate is actually broken down into carbon ba- three carbon counted molecules known as GAP and DHAP. We will get into more details up on this part later. This is just an overview. And the second stage of glycolysis focuses on the conversion of these three carbon molecules, namely as GAP and DHAP, to yield ATP and convert it into pyruvate, which is used further to to yield more ATP. The overall the overall the overall equation of glycolysis is glucose the overall equation of glycolysis can be thought as glucose, which is a six carbon compound, 2NAD plus 2ADP plus 2 inorganic phosphates and plus 2 ATP yields pyruvate, which is a 3 carbon compound, plus 2 NADH plus 2 protons and 4 ATP. So the so the net gain of ATP is 2. Moreover, in the first stage of glycolysis, 2 ATP molecules are consumed, and in the second stage of glycolysis 4 ATP are released. So it can be also thought as the first stage is the energy consuming stage, whereas the second stage is the energy releasing stage. So it is a valid question to think why there are two steps, why there are. Okay. Hmm. Okay, it is a valid question. Why do we need two steps? And what's the purpose of both the steps? So the main goal of step 1 is to trap glucose in the cell and convert glucose into two 3 carbon compounds which can be readily cleaved into phosphorylated 3 carbon units. Okay. So the step 1 is to trap glucose in. So the strategy of this initial step in glycolysis is to trap the glucose in the cell and form a compound that can be readily cleaved into phosphorylated 3 carbon units. Phosphorylated 3 carbon units. So the stage 1 is completed with the cleavage of the fructose 1,6 bisphosphate into two phosphorylated 3 carbon fragments, whereas the 3 carbon units at the end of the stage 2 are further oxidized into pyruvates.